Good evening, and thank you for joining us. My name is Michelle Lampel. I am the director of the Center for the Study of Human Health, and it is my great pleasure to welcome our students, faculty, alumni, and members of the Emory community to this year's Berson Lecture. The Harold and Florence Berson Lecture is presented through the Health Innovation Concentration. This is a degree collaboration between the Center for the Study of Human Health and the Goizueta Business School. The lecture series highlights the intersection of health and business, and it is made possible by the generous support of Emory College alumni and parents, Denise Berson Tanzman and Mitchell Tanzman, who also serves as a trustee of Emory University. Thank you very much, Denise and Mitchell. Tonight's lecture is a tour de force of scientific innovation directed towards life-saving actions only possible through collaborative business efforts. Years before the world had heard of the COVID-19 virus, tonight's speakers were working on antiviral drugs to fight this very type of infection. Dr. George Painter is a professor in the Department of Pharmacology and Chemical Biology at Emory University School of Medicine. He is director of the Emory Institute for Drug Development and CEO of Drug Innovation Ventures at Emory, also known as DRIVE. Dr. Painter has over 30 years of experience in the discovery and development of pharmaceutical agents in the biotechnology and large pharmaceutical company sectors. Dr. Painter was a first-generation college student, earning his bachelor's degree in chemistry, his master's of science degree in physical organic chemistry, and PhD in organic chemistry here at Emory. Dr. Painter has published more than 100 scientific papers and is an inventor on more than 45 patents, 6 of which have led to approved commercially available drugs or combinations of drugs for the treatment of HIV and hepatitis B. He has led international drug development teams, which have generated data for 22 investigational new drug applications and 3 new drug applications. Dr. Dennis Liotta is Samuel Candler Dobbs Professor of Chemistry at Emory and the Executive Director of the Emory Institute for Drug Development. His specialty is organic synthesis and medicinal chemistry. Dr. Liotta's early work focused on HIV therapeutics and the invention of a new treatment approach based on interfering with the way genetic material functions in cell replication. This was a seismic insight and brought about a new era in therapeutics. The drug that emerged from this team, Amtriva, has been taken by more than 90% of HIV patients. This is a very big deal. Over the past 2.5 decades, Dr. Liotta is noted as inventor on more than 90 patents, with discoveries including novel antiviral, anti-cancer, and anti-inflammatory therapeutic agents. He has founded a number of companies over the years to move his inventions along the therapeutic pipeline. His global ventures include nonprofit companies that work on the development of infections such as dengue fever, influenza A and B. And he co-founded Advancing Health Innovations in Africa, an effort aimed at providing the next generation of African scientists with the business and legal skills needed to address their own healthcare issues. Professor Liotta earned his PhD in organic chemistry from the City University of New York and completed his postdoctoral training at The Ohio State University. He was elected to the Medicinal Chemistry Hall of Fame in 2010, the National Academy of Inventors in 2013, and he has received the 2022 SCI Perkin Medal, the highest honor in the United States for achievement in industrial chemistry. Together, the work of Drs. Liotta and Painter have contributed to saving countless lives, and their discoveries are driven by a commitment to humanity and global well-being that is inspiring. Their most recent success occurred when British regulators approved the use of their drug, molnupiravir, for at-risk patients with mild to moderate COVID-19 in November 2021, and it became the world's first approved oral medication. It became the first approved oral medication for SARS-CoV-2, an impossibly marvelous achievement. On December 23rd, when it received the emergency use authorization from the US FDA. As such, it became the 26th FDA-approved drug born at Emory. We are delighted to have you with us this evening, Dr. Painter and Dr. Liotta. During the program, I encourage you all to submit any questions you may have in the chat box. And now, Dr. Painter, Dr. Liotta, welcome. 2 pandemics and 2 drugs. Lessons learned. Thank you so much for joining us.
Thank you.
Okay. Well, thank you. I need to be able to share my screen and it's disabled. So can someone help me with that?
I am not that person, shall I say? Just to keep
Dennis, you should be able to share now.
Okay, well, thank you.
There it is.
Okay, I hope you can all see that. And I'm going to let George start our comments. So
Here we go. Okay. Since over the past 100 years, you guys can see these pandemics that have occurred. Some of them have occurred with great loss of life. And there's every reason to believe, based on climate change, urbanization, all of the pressures on modern society, it's going to occur again. But the point here is, if you look at entry 4, HIV retrovirus, the global deaths associated with that, and you look at 11, SARS-CoV-2, and now it's over 5 million lives, you can see the pandemics that have framed our careers. So tonight, we'll start with the experience with HIV with Dennis and show how that led to SARS-CoV-2 through the development of DRIVE and the Emory Institute for Drug Development.
So you're all aware of the HIV/AIDS pandemic, and there are currently about 38 million people worldwide living with HIV/AIDS. We have tried for 40 years to find a cure, but we've been unable to do that. And the best that we can do is find drugs to control viral replication. And we're going to tell you a brief version of that story, but more importantly, we'll also tell you how we took the lessons that we learned from HIV and were able to apply them to the SARS-CoV-2 pandemic that occurred. And so we'll do this in 2 phases. And in the beginning, I'll tell you the HIV/AIDS story, and George will do what we call color commentary. And then we'll switch about halfway through roles, and I will do the color commentary for his part of the story. So to begin with, the first lesson that we had to learn was to really identify a viral target. Now, for those of you who aren't familiar with drug development, the reason we want to specifically identify a viral target is that there's chances of lower toxicity associated with the drug. If we can hit a target that the virus has, in the best case, that is unique to the virus, then we don't have to worry about disrupting any human targets that are comparable. It's almost never a unique target, but in our particular case, we identified this reverse transcription activity as a target that we thought we could do the best job with in terms of identifying an antiviral agent. And the reason we chose this is because we understood how this process worked. And basically what we were doing in the process is eliminating the elongation of viral DNA. And the way we did it was to add an agent that was like a rail car that was part of a railroad that didn't have a hitch on it on the end to hook up to another car. So this little X that you see at the bottom of the screen on the structure there means that there wasn't a hydroxyl group and we couldn't form this new phosphodiester linkage. So we created an incomplete strand of DNA, which was incapable of ultimately producing viral replication. So that was an important mechanism.
Yeah, no, that's the important mechanism. So you're stopping the flow of genetic information specifically in the virus and leaving the host alone. That's the entire goal of all the classes of drugs we're going to talk about today.
And what's really cool is this was the predominant mechanism, we call it chain termination. But what George was able to discover, as you'll see later on in the talk, was an agent that had a completely different mechanism of action. And this proved to be pretty critical in being able to control SARS-CoV-2 replication without worrying about viral resistance. And we'll talk about viral resistance in just a moment. So the next thing we learned from this virus is that we had to, even when we had the right species, we had to get it to the right place. And in this particular illustration, we're talking about how do we get an entity which, if I just go back one, is highly charged, this triphosphate we show on the lower right, which can't penetrate cell membranes. How do we get it through the membrane into the cytosol and the nucleus where it can actually be part and be incorporated into DNA and do this chain termination process? And the answer was we can't give the triphosphate, so we gave the actual nucleoside, the precursor of that, and we allowed enzymes in the cell that normally phosphorylate endogenous nucleosides to also convert this to the triphosphate and let that inhibit the process. And so this was an important lesson. As you'll see later on when George speaks, the lesson is not just getting it into the cell, but in his case, getting it actually through the intestine so that we can actually produce, get enough material in there to suppress viral replication.
That's right. And so guys, hearken back to Paul Ehrlich, who is the only post-death award of the Nobel Prize, and his idea of the magic bullet, which was around 1910, that actually if you could fire a magic bullet and hit the pathogen and not hurt the host, could you in fact cure an infectious disease? And this is the ultimate manifestation of the idea of the magic bullet.
So we found a compound through some diligent work and some serendipity that had, at the time, by far the best profile of any antiviral agent that existed to treat HIV. And you notice on the top, it says the Emory Wellcome Partnership. And this was a really important lesson to learn. We may be pretty smart in academics, we may know a lot of things, but we can't develop a drug without a commercial partner. And at the time, Burroughs Wellcome Company was the premier site of a repository of antiviral knowledge in the world. And so we were fortunate, because of my personal relationship with George, we were fortunate to create a partnership with Burroughs Wellcome where we could hand off the drug that would ultimately become emtricitabine or Emtreva to Burroughs Wellcome to do the clinical development.
Yeah, remember at the time, probably the modern age of antiviral therapy had been introduced by Wellcome, Burroughs Wellcome, and Trudy Elliott, who won the Nobel Prize for it in 1989 for acyclovir. And Wellcome was privately owned, and all the money it generated went into the Wellcome Trust to help address unmet medical need in the developing world. And our motto was, research is our only shareholder. So while nobody else would touch HIV at the time, we did. And Dennis is right. It's our personal relationship that got us together to help start emtricitabine down the approval path.
Right. So science is necessary, but not always sufficient. And you need these kind of relationships, especially relationships that can get you past some of the inevitable bumps in the road, which We had plenty
A
of.
couple of big ones.
Yeah. So let's address the issue of resistance. And just so you all understand what we're talking about, HIV is typical of many viruses in that there are no proof checking mechanisms for the virus. So about 1 in every 10,000 replications, there's a mistake, which we call a mutation. And HIV has about 10 to the 9th replications per day. So you can imagine just in one day that a whole array of different viral mutants develop. And you can imagine over time, by the way, that these mutants will develop secondary mutations and tertiary mutations. But what happens when we provide a drug to this array of different mutations? What we see is something we call a selective drug pressure. So the drug, if it's reasonably effective, will suppress viral replication in many, but not all of the mutants. So what will happen over time? The mutants that are controlled by the drug, their levels will go down to negligible levels, but the ones that aren't controlled by the drug will just grow back up because there's nothing stopping them, and they will become dominant, and then they will create their own mutants. So this is the problem that we faced, that typically monotherapies fail because we can't control resistance across the entire spectrum.
Right, and that's what happened. So I was on the team that helped develop AZT-retrovir. That was the first drug out of the chute. It was the first drug in this class of obligate chain terminators that Dennis is describing. So after 6 months of therapy, we started to see patients escape from therapy and the viral burden start going up. And I'll never forget this. As long as I live, we went back to the research committee at Wellcome. My then boss, who played a big role in this, a guy named Phil Furman, and we said, look, we've sequenced the viral genome and there are mutations in a critical enzyme, the reverse transcriptase, that lead to resistance, and no one would believe us. Because the hallmark of enzymes that are key in metabolism are generally very conserved. So the idea that you could change one and get resistant was very difficult for people to take at the time.
Yeah. So the way we circumvented this problem was we developed combination therapies. And one of them is shown here. It's called a triplet, and it's 3 drugs. And the important thing about this is that it's 3 drugs in one pill that's taken once a day. And what are the consequences of that? Well, let me show you on the next slide. Oh, I want to say that the development of emtricitabine was hindered by a lot of litigation. Or a lot of our friends in the pharmaceutical sector didn't want competition, so they sued us and we sued them. And it took 6.5 years to sort it out. But fortunately, Emory prevailed in all of these litigations. So one of the important things we learned is speed is important. We can't afford to take 13 years to sort out these problems.
No, I think it was an epic journey. And to say that we were litigated almost into submission is the truth, but we prevailed. And there are a lot of stories around that. But at the end of the day, I think it was one of the most important outcomes for HIV therapy.
Right. You see that Emory made a lot of money at the end of the day, but I always tell people that the real money is going to come from the book and the movie rights.
Yeah, I mean, come on, I'm going to take a couple of minutes to do it anyway. So this litigation came to a head, and I was in the middle of it, of course, and we were in this big room, and it was always filmed, and there were cameras on us. It was very tense. And this lady was interrogating me, and she said, well, why did you become friends with Dennis Liotta? And like I said, there's probably 100 people in the room. I said, I didn't care about Dennis Liotta. And she goes, what do you mean? And I said, well, Dennis Liotta, there was a woman in this lab and I was going to the lab to meet her. And the woman made a big mistake. She goes, well, who is this woman? And I said, well, 30 years later, she's my wife. And so I think, the guy behind her said a bad word. And I think that the whole idea of how we had worked together and how interdigitated we were was part of this, and that teams win. Teams and dedicated people working together win.
And relationships are critical. And that's what we've always had, and what we'll continue to have. So the good news was that between emtricitabine and a related compound, lamivudine, these compounds proved to be enormously effective against HIV. And you see here that there are 18 different combination therapies that contain either lamivudine or emtricitabine that were approved by the FDA. Plus, lamivudine was also the first drug approved to treat hepatitis B virus. So this had enormous impact. But let's not talk about numbers. Let's just show you what this meant to patients. So on the hand on your extreme left, you see there are about 15 to 20 pills. In the mid-90s, this was what HIV patients had to take. They had to take, in some cases, 3 times a day, which meant that they had to get up in the middle of the night to take these. And if they didn't, then their level of protection would drop below a certain threshold and viral replication would occur. Atripla, the drug that we talked about earlier in 2006, combined 3 drugs in 1 pill that you take once a day. So this had enormous impact. And as you can see on the right, over 90% of HIV patients take or have taken this drug.
So, but before you go on, one thing that Dennis didn't say, that the separation of the enantiomers that he did and the identification of what became lamivudine, I was the project leader for Combivir, the one that's on the far left. And we gave, my boss then, Phil, we gave a presentation in a high school gymnasium that was packed. And that's the first time that the death curve started going down in the HIV pandemic was with Combivir.
Yeah, it was remarkable. And so we knew the key: combination therapy for every antiviral. You don't use monotherapy. So remember that at the end of this talk, because George is going to tell you about a drug, an antiviral drug that works as a monotherapy because of its unique mechanism of action. That is what is, to me, so amazingly cool. So one of the things that we learned, remember, speed was important. We learned that the university had a lot of unintended impediments to prevent or hinder drug development in the university. So after much conversation, we figured out a way to circumvent many of these impediments. And we started something we called Drug Innovation Ventures at Emory, which is a wholly owned subsidiary of the university. But because it's a separate legal entity, it isn't subject to the same rules and regulations of the university, which meant that those unintended consequences, unintended impediments that existed in the university, we were free of. And when George agreed to be the CEO of Drive, I knew we were destined to do some great things.
I got hit in the head that day and was delusional.
Yeah. So let me just give you a very quick idea of what Drive is. It's, I said, a wholly owned subsidiary, but the important thing was that it was led by an experienced team, people who had lived drug development. So I know a lot about drug development. I've seen snapshots of drug development, but I haven't lived it. George was in the trenches, and it was essential to get somebody who knew how to do this. And that made all the difference in the world. And because of the network that we created, we were able to bring drugs through the early parts of clinical trials through what we call phase 2A clinical trial, the first efficacy studies. And we created a separate entity called the Emory Institute for Drug Development, which is the research arm. So you can imagine DRIVE as kind of the development arm and the Emory Institute for Drug Development as the research arm. And the two were intimately intertwined, and that's what made this operation work.
Agreed.
And George, I'm going to turn it over to George now, and he's going to tell you a little bit about how government action played a role and then how we were able to move this thing forward.
So really one of the drivers, starting back in 2000, I'd become very involved in biodefense and the idea of finding countermeasures for biological attack. And the government, the US government, began a series of actions to fund research and development for the development of these countermeasures, starting with the BioShield Act in 2004 and the Pandemic and All Hazards Preparedness Act in 2006. And there are many more after that. But a lot of money was set aside and administered through Health and Human Services to fund efforts like DRIVE and the EIDD. So we knew we could finance our effort to address these viruses. Next. So you guys know where this is. Right now, this pandemic has been going on and on and on and on. But we were ready for this because almost 20 years ago, started looking at RNA viruses. These are viruses that carry their genomic information as RNA. There are over 204 identified RNA viruses that cause human disease. And at any given time, they're probably responsible for up to 80% of the global disease burden. For the vast majority of these, there have been no effective drugs. So there's a list, you can read it, but you'll see coronaviruses on there. But the program that ultimately led to molnupiravir started with toga viruses and Venezuelan equine encephalitis virus, VEEV, which is on the second line.
Yeah, and let me just interject that back over 10 years ago, George and I had this discussion and we came up with this strategic vision. Imagine if 80% of the viral disease burden worldwide are from single-stranded RNA viruses. It seems obvious that if you want to be prepared for the next pandemic, you would find a way to try and address them, particularly if you could find a compound that had activity against multiple viruses. Then you're increasing your chance of being in the right place at the right time. And that's the vision that George had that led us to where we are now.
So, okay, guys, a pandemic, a public health emergency, or in our case where we started, I was contacted in 2013 by the Defense Threat Reduction Agency looking for a medical countermeasure to treat VEEV, Venezuelan equine encephalitis virus, that had been weaponized and was potentially usable against warfighters, soldiers. But you developed something called a target product profile that describes the ultimate profile you want for a drug for that sort of situation. And not unsurprisingly, the overlap between what a warfighter might use under combat conditions and what a civilian might need in a pandemic, they're very close. You want something that's stable. You don't need a cold chain. It doesn't need to be kept at -20. People can take it themselves. It can be delivered to their door, believe it or not, by FedEx was one of the national strategies. And you take the pill. It's very bioavailable. You don't, if your diet or your state of health varies wildly, you're not going to have to adjust the dose. And you can read the rest. And it's distributed to the appropriate tissues for the disease. So interestingly, here we go. Our goal was to develop a drug for VIV that had been aerosolized. It had been made so it's flocculent. It could be put in a circulating air system, an air conditioning system as a bioterrorist act, or it could be sprayed over a battlefield and incapacitate soldiers. So the virus is breathed in on inspired air, and it binds to olfactory receptors deep in the nasal passage, and then is moved up the axon of your olfactory nerve, which isn't synapsed, it's not broken, goes right through the cribriform plate in your skull into your brain. And in 18 hours, you have debilitating encephalitic disease. And in addition, it caused interstitial pneumonia because you would inhale it. So you can see that the idea of having CNS involvement like coronavirus does, and that's probably one of the reasons you get long COVID that's associated with cognitive impairment, or you get these pneumonias and you get pulmonary damage. So all of those factors that went into designing a drug for use under battlefield conditions actually turned out to be relevant to developing a drug for public health for coronavirus. So I'll go over this very quickly, but you need the drug to get into the brain quickly. And there are actually very well-defined, well-studied transporters in the blood-brain barrier, the barrier between your blood and entry into the brain. And one group that's transported very effectively is uridine. It's in the middle there. So we started looking at analogs of uridine as potential countermeasures for VIV. And that led us to the drug on the left, identified here as EIDD 1931, N4-hydroxycytidine. And what's interesting about this versus the work going back to HIV is this is a ribonucleotide. It has a hydroxyl group, two of them, one in the 2', one in the 3' position. These compounds turned out to be very, very broadly active. Unlike the compounds that were a big struggle, like lamivudine and emtricitabine, they were active against HIV and HBV. But here you can see, and one of the group of viruses that early on we knew it was active against was SARS and MERS. So when SARS-CoV-2 came around, we were ready. So Dennis spoke about mechanism of action, and this one is very unique. So if you look in the panel to the left, the gray panel, you'll see two forms of the drug. The one on the left, on the bottom, is an oxine, and it has two lines, a double bond between nitrogen and the C4 carbon of what would normally be cytidine, and a free hydroxyl. And the little red circle is a lone pair that acts like the lone pair on the carbonyl of uridine, so it can look like uridine. And if you go on the left, simply by moving the double bond, a process called tautomerization, it looks like an amino group, so it looks like cytidine. So what happens, go over to the right, you're looking at coronaviruses are called positive strand RNA viruses. So they have this special kind of orientation of their genome, their RNA. So you want to make another copy, you have to make a full-length negative chain first, and then that's the template for a full-length positive chain. So if you incorporate, and Dennis talked about the 5' triphosphates, you have to make the 5' triphosphate. That's done inside of the target cell, and it's incorporated, say, as cytidine on the right in the gray box in the first strand, and then it tautomerizes to look like uridine in the left box, and then you make the negative strand, it will introduce a mutation called a transition mutation. And the introduction of those mutations in these RNA viruses pushes them over the edge of the ability to live. And so they lose their viability. It's called error catastrophe. And that is a very, very unique mechanism. So what happens very quickly is the virus accumulates mutation because of this process, polymerization, and boom, there are too many mutations and it can't work anymore.
So I just want you to realize how really clever and critical that mechanism of action is, because it allows us to control viral replication with a single antiviral agent, not 3 drugs, because we don't have 3 drugs to do this.
No.
This is one drug that works effectively. And that is, to me, the most magical part of this discovery.
Yeah, Dennis pointed out, and I'll go into this in a minute, that we're science geeks.
Yes.
Sorry, we can't help it. So the idea of catastrophic mutation, which is very involved virology, didn't come from virology. It came from physical chemistry, from Manfred Eigen. And it was phase transition, first order phase transition. So when ice melts, it does it by a cooperative process that's concordant across the entire matrix of melting. So if something happens here, then way down here it's felt. So in the viral genome, which is rather big for an RNA virus, there are 37,000 bases. Something happens at base 10, base 200 may feel it. So very quickly, you accumulate enough mutations that it destroys viability. So one of the things that happens because of that is theoretically it's impossible to get resistance because it collapses too quickly. And this is just a slide full of publications from us and our collaborators showing that's in fact the case. We could not get resistance to the drug that became molnupiravir. So a little bit of a wrinkle. So you go back and you look at the compound N4-hydroxycytidine. And I think this is one of the triumphs of what we put together, that we have experienced drug developers proximal to innovation. So we can iterate. We don't innovate, give it away to the developer and have them develop them and go, uh-oh, it doesn't work. We just feed back to each other. So on the left of this screen, you see molnupiravir. And if you look on the far left, you'll see a little piece of isobutyl ester. That was put on there because the original molecule, quite unanticipatedly, was trapped in the enterocytes, those cells that line the intestine that are responsible for absorption. The first biological barrier the drug sees, it was quickly turned into the triphosphate that Dennis described. Well, then it was trapped there. So we had looked at various species, including dogs and ferrets and all other animals for various reasons. But in non-human primates, the bioavailability dropped down to 4%, way, way, way below. And I've seen this before. We knew it was trapped in enterocytes. So we started out with EIDD 1931, but EIDD 2801, simply covering up that hydroxyl that's phosphorylated long enough for it to get in the blood, led to a drug that was usable.
And this was a strategy that was established years before at Burroughs Wellcome with making acyclovir, an orally active drug of valcyclovir. So it was well precedented, but this is where experience actually plays an important role.
So how did all this happen? So that's where the story starts to accelerate and goes sort of to the business side, but affects the science side. So 2018, we identified 2801, and we were on track in 2019. And by that time, we had about $30 million from federal contracts, and we were developing. We were going to open an investigational new drug application for flu and continue to work on VEEP because you can't study VIV in people, so you do it in animals under what's called the FDA animal efficacy rule. So we're going to get an IND open, do phase 1. So then go to 2019. By January of 2019, Chinese scientists had identified a novel beta coronavirus as the causative agent of a cluster of 2020, sorry. Yeah, well, I don't know what day it is anymore after molnupiravir. So 2020, they had identified the beta coronavirus. So I was at the JP Morgan Healthcare Conference and received a call from agency at HHS, from the FDA, and from the National Institute of Allergy and Infectious Disease that said, hey, that drug you're working on potentially can treat SARS-CoV-2. Are you up for this? And we said, of course we are. So we quickly found a commercial partner in a biotech, and we accelerated the whole process. So I think if you look from the top to the bottom, and the chief medical officer who helped on this is sitting off screen to my right, and she can disagree, or agree. We did, I think, in 18 months, 4 years of work. And regulators in the United States and the United Kingdom were on the phone with us 24/7. So very quickly, the drug got moved into phase 1. But there was animal data in the beginning. And I don't want to go through all this except to say, if you look at the blue boxes in these histograms, those are viral burden, SARS-CoV-2, in human lung tissue that's implanted in a mouse. And then on the right, you can see in the yellow box, after treatment with what became molnupiravir, EIDD 2801. And very quickly, and then on the far right, under D, is the composite of multiple experiments. So we were like, oh my God, you get 6 logs drop in viral burden after dosing with molnupiravir. And that was profound and unanticipated. So then we had to deal with something called weight of evidence. So if you look at the mechanism, the drug causes mutations in virus, and it had a positive response in what's called the Ames assay, which is a very rudimentary way of trying to see if something will cause problems in humans. It was positive. And so we had a lot of biochemical insight that suggested that it was peculiar to bacteria. So by that time, we were partners with Merck and Ridgeback, Merck, and Drive all worked together. And the weight of evidence in vivo in these experiments, which you can read, said it's not mutagenic. And a lot of data has been accumulated since then. So going back to what Dennis said, if you can reach into an organism and touch the virus and not touch the host, that's called specificity. That's what you want to do. And I think people were prepared for how specific molnupiravir was, but there was a lot of biochemistry that led us to believe that was the case. That is the case. So into people we went. This was run by Ridgeback. Next slide, Dennis. And this is what's called a phase 1. And when you look on the right, that's where you give it to people for the first time. And it's a safety, tolerability, and pharmacokinetics. You want to make sure enough drug is on board to treat the disease. The pharmacokinetics were beautiful. So if you gave someone 200 milligrams and you gave them 800 milligrams dose, the exposure was 4 times higher. It was perfectly linear, and there wasn't a great deal of variability. And the safety officer is sitting over there, but I think it's fair to say it was very safe and well tolerated. Is that fair? Yeah. Without getting in trouble with,
with
the FDA.
And this is not the norm where you see these linear pharmacokinetics. So
No.
This is very fortuitous, and you can't really easily predict that the PK is going to be linear. but we were fortunate that it was.
Yeah, I've done a lot of these over my career. That's the first one I've seen like that. And I think the phase 2 is where you actually look at efficacy. And that phase 2 protocol was designed by Wendy and the colleagues and us helping them. And what was really profound is underlined, that the dose turned out to be 800 milligrams twice a day, BID. And that by day 3 after dosing, all but one patient out of 53 in the phase 2 had no virus on board. And there was a dose response that showed at lower doses, the efficacy changed with dose. So the choice of 800 milligrams BID was validated. Merck carried that into phase 3. And interesting, if you look down on day 5, none of the participants in the molnupiravir 400 milligram or 800 milligram groups had virus. So I think it's very clear, and subsequent data shows that probably by 36, no later than 48 hours, viral burden's driven down to baseline, that it hammers the virus and the virus is no longer viable. So which one is this? It's hidden. So the phase 3 first one was done at Merck. Everybody who follows this knows that the first indication was 50% efficacy, the second 30. And there was a great deal of discussion over this, but meta-analysis and work in India and work by WHO at doing the meta-analysis show that it's much higher. What, probably 60? And so what happened? And I think what happened in the second part is the scariest part of all of this for me. I've been doing biodefense for a long time. And I think we can do the kind of work we're doing very well. But when you need to run a clinical trial against a background of a pandemic, that's where the problem lies. The presentation of the virus clinically is polyphenotypic. Remember, people had blue toes, people couldn't breathe, people had to be intubated, people had heart attacks. There's all kinds of manifestations. And over the course of therapy, not only did the patient population change, but the virus changed. Remember, it started getting resistant to monoclonal antibodies, et cetera. So it wasn't a reflection of efficacy of the drugs. It was a reflection of the changing background against which the drug was tested. This is going to be the biggest debate in going forward. How will the physicians construct clinical trials to show efficacy? I think that's our biggest challenge. So at the end of the day, the first approval for emergency use came from the UK. We worked very closely with the UK. The Ridgeback team was on the phone with the UK night and day. I think it was probably the fastest drug approval I've ever seen. Molnupiravir has been approved by or endorsed by the WHO, and it's in use in over 140 countries around the world. We're starting to get data that shows that it really works. So I think the whole thing here is you can respond quickly with the right people. You can respond quickly with cooperation with regulators. And I think that I said to people before the show started that what was unanticipated is all of the voices that come in the modern world through social media and internet, and it strongly influences how things are conducted. So I think in the future, you're seeing things like the AVID program, antiviral drug discovery initiative for pandemic preparedness. We're going to be part of that to the tune of probably 100 million a year. You saw Operation Warp Speed, which I think is probably the most profound development in antiviral drug discovery and development to date, and that's going to influence how we go. So that's our next challenge, to integrate ourselves into this new paradigm. And that's why we need all of you. We need to train everybody, and we need the next generation of drug developers here. So
we're not done yet.
we're not done yet.
And we need your students to come forward and join in the initiative. So thank you very much for your attention. I know we went a bit longer than intended, but I think we had a lot to say. So thank you very much.
Thank you, everyone.
Thank you so much. You did not go too long. This is absolutely inspirational, and it's the call to action is serious and energizing. Let us now thank you very much and open the floor for questions. Kate. Do you want to read from the chat, please?
I sure can. So from Ryan Sayed, and I'm sorry if I get your name wrong. What are some ways in which stigmas around HIV and the politics around COVID-19 influence the development of these respective treatments?
Oh my God. I think that's another hour-long talk, but thank you. That's a very
relevant question.
Very relevant question. So we, the stigma with HIV in the beginning, with all the opportunistic infections that they had, it became obvious to many people that these folks were HIV positive, and they were, of course, shunned. It was just outrageous. And then later we found, after long-term use of HIV drugs, that some patients developed what we call lipid dystrophy. They had fat distribution, so some would even get humps on their back, or they would get a thinning in their face, a narrowing, and all of these things were caused them to be stigmatized. And better drugs actually tended to minimize the stigmatization. How we deal with politics, if anybody has a really good answer for that, I'd love to know, because it's very debilitating and causes tremendous distractions.
Look, on both those questions, I was at Welcome for AZT and was the liaison to ACT-UP, the AIDS Coalition to Unleash Power. And these were generally at that time identified as men who had sex with men, and were, I think at the core of your question, were socially stigmatized. There was no question that was true. But these were very highly educated people in a lot of ways, and very vocal and very involved in treatment therapy.
Thank God.
And well, it was good. And sometimes they could see through the blinds on trials. So I think they had a big influence on expediting discovery. And then, of course, it broadened into hemophiliacs. It broadened into other people. It broadened into the heterosexual community. So the stigma got a little bit diffuse.
But there's your answer, don't you think, really, which is it was the activists that actually changed the way AIDS therapy finally developed. They got the attention of government provided funding. And so it's the responsibility, I think, of every individual to make their voices heard and to say, we need more science, we need more training, and because that's the only thing that will actually save us in the long run.
Yeah, I think the politic question is an interesting one too. I ducked politics. I was asked to be the Assistant Secretary of Preparedness and Response and be in charge of this kind of thing. And I think what was unanticipated is how drugs were used as tools for political agenda. And I think picking sides and Instagram influencers and people like that who I would never have anticipated to have such an impact. And I'll tell you what I think about that, frankly, that people spoke out with various agendas ranging from wanting attention to using government support of a drug to indicate some sort of political indication. Well, you know who the victims were? Really, in round one, I think the drugs could have saved a lot of people in long-term care facilities. And I think a lot of people died because of these agendas. And I think somehow going forward, we got to get past politicizing every decision associated with health to the benefit of the general public. I don't know how to do that yet, but I think that's what we need to do.
Wonderful. Thank you. From Amna Soniwala, what was your biggest challenge in conducting this research? How do you stay motivated during such a draining project?
Well, you know, it's not a job, it's a passion. And I think we were caught up in COVID-19. We took a lot of body shots on COVID-19. There were whistleblowers claiming we tried to get federal funding inappropriately. There were internet influencers saying the drug was going to turn everyone into zombies because of mutations. And I think that's when you put your head down, you keep your eye on the mission. The mission was to save people's lives, and to do that in a responsible fashion, interact with regulators. And we were supercharged by all the people around us, the healthcare providers. Everybody was looking for a way to address this. So I think there was a lot of energy in the community. Ask me right now, I feel like I've been beaten up for 2 years, but we'll be ready. We'll be ready for the next one.
So you have to be smart. You have to be innovative. But even when you're smart and innovative, it's not quite enough. You have to be resilient. When you get knocked down, which you will, you got to stand back up and go forward. And you got to do that time after time. And that's what we're pretty good at. We get a little perturbed. Occasionally, we might use some foul language, but we go forward. And so resilience. Don't ever forget about resilience, because without it, you don't get anywhere.
Yeah, and I think I have a picture of myself sitting on the steps of my son's house when I got a call that the drug had worked. And it still gets me. I still almost choke up. And I immediately got a call from Dr. Lee, Dean Lee at Merck, who is a very famous cardiologist who took on the job of president of Merck Research Laboratories globally. And we were both in tears. And he told me, when was that? March, February? That his mother had died of COVID in January. So I think people have this vision of pharma as this monolithic, uncaring entity. And indeed, there are some aspects of it that may be. But again, you see the dedication of these people and their concern and their involvement, and it changes everything. It just changes everything. It's not a job anymore. It's a passion. It's a pursuit.
That's great. Thank you for sharing. I think that's advice that we can take whatever our path in life. From Carolyn Hahn, how was your undergraduate experience linked to what you do right now?
Interesting.
Well, you mean beyond the beer and intramural
that's
sports?
The problem. Maybe it was in a bizarre sort of way, because we both had somewhat similar undergraduate experiences, which were, let's just say, not as serious a pursuit of knowledge as we might have wanted. And in my case, that actually turned out to be a very good thing in a bizarre sort of way.
Very bizarre. I want to hear this.
Yeah, because I was clearly, my academic credentials were deficient, and I knew that I had to work really hard to try and catch up. And I did that in graduate school, and I did that as a postdoc. And then when I came to Emory, I realized that I was competing against a lot of people who had much better pedigrees than mine. So I had to work really hard to compete and in some cases outdo them. And that actually never left me to this day. I still work very hard. And so I don't recommend my approach to anyone else. So please academically do the very best job that you can. But ultimately, I talked a little bit earlier about resilience, but it is that drive, that belief that you can get out there and you can make a positive difference. That is our driver. That's what gets us up every day and gets us going. And so you're very fortunate to be at Emory, where you have a wonderful opportunity to learn many different things from many different wonderful people. Just enjoy that opportunity, take advantage of it, run with it, and don't be in a position where you have to always compete. Just compete naturally, because it's a great thing to do.
Yeah, you know, I mean, I agree with everything Dennis said, and I had this wonderful experience. You know, I was in Central Asia and Siberia, actually Russia, and in a university, a little one down in between Mongolia and Kazakhstan. And we were having dinner and everyone had a toast. And it was interesting what came out of my mouth, because I think it bears on unfiltered vino and veritas, or veritas and vino. But, you know, I was interested in science. Science was my driver. And so what came out of my mouth was, well, you know, I'm here with you in a place I would never have contemplated in my life being. We were working on tick-borne encephalitis, which kills a lot of kids. And science has been a magic carpet that brought me here. And I think what you're learning now and your experiences now are really the basis of what's going to drive you. So yeah, we weren't the most serious. It wasn't the most serious times. Then I think times are more focused now. Dennis watched Animal House over and over and over again. I just think that you're learning now things that are going to form the basis of what leads you to the next step. And he's right, persevere.
Favorite college, knowledge is good.
And the dean has unlimited power. We already know that.
Yeah, we're not
Okay.
gonna do that.
We're not going to do that.
Kate.
Yes. Well, I think you just reassured a lot of undergraduate students. If you all have time for a couple of more questions, we'll roll straight on through. From Victoria Lashak, what advice would you give for future drug developers researching RNA virus drugs such as HIV?
Well, you know what? Yeah, I just got a whispered from the CMO, who's also an Emory chief medical officer, who's an Emory alumni. She's a physician now, but was Dennis's student. Look, I think there are a lot of ways to go here. I'm really driven by what are called mega trends. So you can go science, you can come over and see us. That's what we do. I think, honestly, right now, we're probably the best in the world at this. But there are things like climate change, there's urbanization, there are all kinds of social pressures, there are all kinds of public health issues that are going to exacerbate conditions that lead to the emergence of these pandemics. So think about the direction you want to come at it from, and then just go looking. There's a lot of it in Emory. There's a lot of it in the world. There's a guy I love called Mark Woodhouse at Edinburgh who does all the epidemiology and all this issue of 2 new viruses a year and how these viruses evolve, that all comes out of there. So there are a lot of people around the world addressing lots of aspects of this. So look where your general interest lies. And then, yeah, call Dennis, all right? We can help too.
Yeah, I always tell students, irrespective of all the things you may have been told by your parents, by your relatives, by your friends, all well-intentioned, you have to make your own decisions about your career. So find something that you're very good at and something, hopefully the same thing that you're passionate about. And I can guarantee you that if you're good at something and you're passionate about it, you will be successful. And probably you will even find a way to make money. So just take money out of the equation, follow your passion, and you will make a difference, a positive difference in the world. And that is the best legacy you could ever hope for.
Right. And I think it starts at the one person level. I think for me, the thing that really impacts me the most is when we get emails that say, you know, I was dying from COVID. I'm old. No one was interested, and molnupiravir saved me, or Combivir, you know, saved my significant other, those kinds of things. So it starts at the one person level and goes. So I just want to say that to support what Dennis already said.
It's wonderful. Thank you. From Kate Siegel, were there any ethical implications in terms of the drug development, such as the trial period in which it would need to be tested on human subjects?
Yeah, you know, as again, we've got the experts sitting around the table. The issue on ethics and conduct of clinical trials revolves around informed consent. So the informed consent forms on the early HIV drugs and the informed consent forms on molnupiravir were way over the top. Any possible issue that might be associated with participating in the trials was identified, and it was made clear to all participants what those were. So based on a well-constructed informed consent that went through multiple regulatory agencies, you can rest assured that the patients understood absolutely what any risk associated with participation, what risk there might be.
Yeah, so it is, it's a very complex issue, and we continue to learn about how to communicate with patients. And in a pandemic situation where hospitals were overwhelmed, it gets obviously much harder to do because people are just doing what they can do to stay alive. And so it's a very complex and very important issue. And I hope that now that we're past this acute stage of viral transmission, that we can maybe step back and take a hard look at what we did and develop some best practices going forward.
Yeah, I don't have any concerns over the ethics.
Right.
What I have concerns over are how do you accelerate things under dire conditions? So let me just tell you some of the questions that came up during the course of coronavirus outbreak based on biodefense. Look, not everybody in the world likes everyone else. We all know conflict, and we're witnessing barbaric conflict that we never thought we'd see. So what if in the middle of coronavirus, some non-state terrorist group introduces another RNA virus, and the symptoms of that infection onset are masked by coronavirus? And then all of a sudden you realize that, oh my gosh, we're not dealing with what we think. And then you have to deal with risk benefit, right? There's a terrible risk. We have to provide benefit to the patient. There's informed consent. There's how do you handle all that? And so as we go forward, respiratory disease is probably going to be the next one. It's going to happen in urban centers. These urban centers are crowded and they're correlated with low GDP, gross domestic product, poverty. Okay. And so these things start to emerge. And then if somebody chooses to use that as an opportunity to mask an attack, how do you handle all this? All this stuff is in the mix, and all of it's stuff that we're going to have to learn how to deal with, and we're going to have to learn how to deal with it quickly.
And I think one of the things that people don't want to hear, but need to hear, is the WHO needs to take a critical look at itself. The CDC needs to take a critical look at itself and see if what they're doing is actually the best mode of action, because I'll bet it's not.
Yeah, I think that's true. And you know, over the years, I've been, I don't want to say forced, but to the questions earlier about how do you become involved in this? Somehow I became involved in the business of this. And that started out at Wellcome and talking to analysts on Wall Street. But in the middle of all of this, there's huge costs associated with this. Most of those costs are going to have to be borne, at least at the early stages, by government. The United States has done a very good job of that, and that's why we have DRIVE. But governments are politicized by virtue of it, right? And the earlier question, how do you get around the politics? I think the answer is that you don't. And that somehow the WHOs, the CDCs, somehow we have to come up with a structure whereby they can do their job regardless of pressures that might result from polarized politics or aberrant voices that come in from the internet, et cetera. So I think we have a big challenge ahead of us. It's not just what Dennis and I do, but it's social issues, issues of risk benefit, even issues of government, because Forbes identified last year one of the biggest risks to global order is the growing lack of confidence in governments. And I think we've all seen that. And as that confidence erodes and populations refuse to abide by government dictum, that's going to be fertile ground for pandemics. So there's a lot of room for you guys to do work, okay? We need you.
Yeah. Well, and in follow up to that, if you don't mind sticking with two more questions, Yuki Miyatake asks, what's the role of academia in work like this and drug discovery?
So I've spent my entire life trying to show that, or my entire professional life, that academia is not the ivory tower, that we have smart people who aren't just interested in esoteric, unimportant little problems, but that we can be part of the solution to address important social issues. And one of the things that I'm most proud of is that I got a terrific guy from the commercial sector interested in moving to academia and working with us. And so this is one of the things that I want to see at Emory, is that we bring in more people from the commercial sector, because I think it will be a positive cultural change. It will enable us to use the vast knowledge base that we have and to pair it with the realities of the commercial sector, the business world, and allow us to provide real world solutions to real world problems.
Agreed. I think that's it.
That's great. Thank you. And one last question, which I think is lovely on which to end, from Skylar Lewis, the process of pharmaceutical development is not an easy one, full of rejection, trial, and error. How do you decide when to celebrate your creation, discovery, or work?
Yeah, well, my wife, the CMO over there, is laughing. I think that is a good question. And so I'll just tell you my personal history. My boss was Trudy Ellian, and like I said, she got the Nobel Prize in 1989 and really was the catalyst for a lot of the work we do. And I was much younger. She called me into her office one day and said, look, your job is to go out there and take this discovery, and I don't want you to come back into my office until you have in your hand a pill that works and helps people. And so I did that for 16 years, and I was on airplanes all over the world. And when one job got solved, there were 10 more to do to keep the drug moving. So I never learned to celebrate. And I regret that. And I think I don't know when to celebrate, but thank God there are people around me that do. And I think I celebrated the day that Dennis didn't go into it, but one day we got information that M-tricytamine, and by the way, that was named for Emory Triangle, the biotech company we were all running, and cytamine's the class name, that resistance came very quickly. And that observation led to Combivir. I think I celebrated then. I celebrated when Combivir was approved, and then it was a blur. And I think I really celebrated with that molnupiravir worked.
Yeah. So I used to say all the time that the viruses keep us in business. So we have some success, but then another one of these guys pop up. It's a whack-a-mole kind of game. But I celebrate when I see the students that I interact with commit to being part of the quest, going forward, making a positive difference. To me, we may play whack-a-mole for a long time, but the more people we have in this game, the better. So you folks out there, you are the future. You have the opportunity to make a really big, important difference by using your mind, by using your passion, by using your business skills to change the world. And my celebration comes through you. So, and I'm gonna
Cheers to
say,
you guys.
yeah, cheers to you guys. So thank you very much.
Let me say one thing that was really interesting on the business side is, so I got the call from Ridgeback and then from Merck, and I think my wife knew before I did, but she was closed-mouthed about it. But at any rate, that the drug worked, and that was released in a press release. In the immediate hour after that, value of the global markets went up by 8%. So there was almost $100 trillion change in market value on the announcement that there was an oral antiviral. And I had this like, you got to be kidding me moment. And I think every now and then there's a very graphic indication with indicators that aren't so hard to see of what all this means. So I don't know if I celebrated or was terrified at that moment, but I'll take it as a celebration.
So thank you all.
Thank you.
And we celebrate your work, and thank you very much for everything you do. It's been a wonderful evening. Thank you. Thank you very much.
Thank you, guys.
Thank you.
Good luck. And if we can help, we're here.
Lovely. Good night. Thank you.
Bye.
Bye-bye.